And we are recording. Hello, students. My name is Brendan, and today I'm going to be presenting on viruses and giving a brief overview on what exactly they are and how they're classified, and then going into why exactly we should be studying them, why they're so important, especially now more than ever. So to first start off, viruses are submicroscopic. As you can see from this image, they're smaller than most other beings of their type, especially most bacteria, for example. That's the closest thing we can really get to. And so most of you students have probably taken a biology class by now that has allowed you to use a light microscope to detect probably some kind of paramecium or something. And so you're probably already aware of that even bacteria can sometimes be very difficult to detect. Well, most viruses are essentially impossible to detect with light microscopes. They require a more precise, specialized microscope called electron microscope, which is usually otherwise reserved for like subcellular compartments. So that should really speak to how small most of these pathogens really are. As you can see from the title, I labeled viruses as nanomachine pathogens, and that's because there's somewhat of a disagreement of whether viruses should be considered living or not. The general consensus is that viruses are not living, and there are a few arguments for that, but there are a good number of respected scientists who say that no, viruses are more closer to living things than we really should give them credit for, that we do give them credit for. This first argument right here, let me just bring up my pen. This first argument here is that they're non-cellular. That's an argument against viruses being living. However, despite this, a lot of scientists actually hypothesize that viruses evolved from cells and that technically we all share a common ancestor with viruses at some point throughout the course of evolution. So that means millions and millions of years ago, viruses might have branched off from the rest of us living things. And so really that could be an argument in favor of viruses, that they are derived from cells. Another argument against viruses is that they require hosts in order to complete their life cycle. So as you probably have already learned in your classes, there are other types of parasites. There are parasitic worms, there are parasitic fish, there are parasitic fungi, bacteria, etc. But most of these life forms can still complete their process, their life cycle by themselves, and that they kind of just exploit their host for extra resources. However, viruses cannot, absolutely cannot replicate their genome. They can't survive really on their own. They need something else. They need some other host in order to produce progeny and to produce babies, basically. An argument in favor of viruses being considered living is their structure. They're made of genomic material, either DNA or RNA, surrounded by a protein shell or a capsid, otherwise known as a capsid. This is one of the fundamental principles of biology. One of these fundamental principles is that living things are comprised of genetic material, usually DNA or RNA, that code for proteins. And if you haven't already learned this in your bio classes, proteins are basically magical molecular machines that are responsible for almost anything that you can think of when it comes to the cell or the body. And the fact that viruses have DNA or sometimes RNA, that code for protein would, put it closer to the rest of us living beings. So that's an interesting thing to keep in mind. And finally, another argument in favor of viruses being living things is that they evolve. Non-living things don't evolve. This is an interesting observation because your vacuum cleaner doesn't evolve, your shoes are not going to evolve, and despite what you might hope, your pet rock is not going to evolve either. But viruses do evolve, and so this is possible evidence that viruses are indeed living beings. So those are some of the defining traits of viruses and what kind of pushes and pulls them toward either category of living or non-living. But going on to exactly the proliferation of viruses, there are 219 viral species known that's just known to humans. So there's probably a lot more viruses out there that affect humans that we just haven't discovered yet. And on top of that, humans obviously are far from the only species on Earth. And so once you account for the millions and millions of other, insects and mammals and bacteria, fungi, plants, etc., you end up with an estimated hundreds, let's underline this, hundreds of millions of viruses on Earth. That's insane. These viruses, or all viruses, have some kind of either direct or indirect impact on essentially every organism on this planet, and that's amazing. That's something that we cannot forget. These things are really important. If there are no questions so far, I will be moving on to the next slide. Let's just reorient this here so you guys can see better. I want to talk about how we classify viruses exactly. So there are a few ways. We can either use genetics. Let's circle this here. We can use structure, method of infection, and so on and so on. So this first method, genetics, let me switch to my laser pointer. There we go. So if you can draw your attention over here, like I mentioned, viruses are comprised of either DNA or RNA, but that's not the only defining trait of their genome. So as you students have learned in your class, human genomes are made of double-stranded helical DNA. It's basically like a ladder with rungs twisted up like into a double helix. However, that's not the only type of genome. So this would be human genome. That's not the only type of genome that exists. Viruses can be double-stranded DNA like us. They could be single-strand DNA. They could be single-strand RNA, or they could be double-strand RNA, or they could be a mixture of both. It kind of gets a little more technical, and I will avoid the jargon for the sake of this presentation, but you should keep that in mind. The main takeaway here is that viral genomes can take a multitude of different forms, and that's a way that we can classify them, is their genetics. The next way we can classify them is by their structure. Viruses, like I just mentioned in the previous slide, there are hundreds of millions of different viruses. And so obviously, even though they have a lot of unifying principles, there's going to be a wide variety of diversity as well. So if I could just get a clearer picture here, here are a few examples of the way viral structures are labeled. They can be, they could be cylindrical, they could be icosahedral, they could be helical, they could be complex. It depends, and depends on how their membranes are composed or the interactions between their proteins. As you can see, there are a few examples of each of these, like bacteriophage is probably the classic example you're going to see, especially T2 virus. I'm not sure if you guys are familiar with that. HIV, I know a lot of you know about already. That's an example of a more cylindrical virus. And then there's influenza adenovirus, so on and so on. So that's another important way that scientists classify viruses that's very convenient in certain circumstances. The next way we can classify viruses is how they infect cells. So we all know viruses infect cells, but how exactly do they go about that? It's just like all of us students, we go to school in the same way, at least when it's not virtual. We all go to school in the same way, or not in the same way. We go to the school ultimately, but we go in different ways. Some of us drive our cars there. Some of us who do drive our cars will park on campus. Others will park off campus. Other students will walk to school. Others will get a ride from someone else or take their bike. That's analogous to all of this. So viruses are ultimately going to infect the host cell here, the intracellular space. But it's a matter of how they penetrate this membrane, this outer line right here. Is it going to be clathrin-mediated endocytosis, caveolin-mediated endocytosis, or is it going to be independent of either of those proteins? Is it going to be more of a direct fusion into the protein? Now, again, that's some jargon I'm tossing at you. All you need to know really, for the sake of this presentation, is that clathrin and caveolin are different types of proteins found on the surface of membranes. And when viruses bind to these surface receptors, that can facilitate entrance into their hosts. So that's another important way we can classify such pathogens. And then I included this image as well, this GIF, just to kind of quickly exemplify how complex viruses can be. And so this is obviously a partial explanation for why there are so many different ways we classify viruses, is because despite the fact that these things are submicroscopic, despite the fact that they are fairly simple, relatively speaking, they are actually very complex in many ways. And finally, on a final slide, I wanted to talk about some of the pertinence that viruses have. They have worldwide relevance. They have, like I mentioned earlier, they affect essentially all organisms on the planet, whether it's directly or indirectly. So there's a constant flow of research and articles coming out dedicated to viruses, but I decided to choose a few in particular, 3, one here, one here, and one here. So to focus on this article first, this one was investigating the immune trait that could be responsible for making a person more susceptible to SARS-CoV-2 reinfection. And as many of you guys know, SARS-CoV-2 is the virus responsible for COVID-19, the pandemic that we're currently in. And so, as you may have heard, once you're infected with the virus, you produce antibodies, you're good. Not exactly the case. You might be good for a while, but you may be susceptible to reinfection later on. Viruses, again, very complicated sometimes. So what this paper was studying, it's an emerging scientific paper, and it seems to suggest that individuals who have lower levels of antibodies, these proteins that are responsible for essentially killing invading pathogens, if people have lower levels of antibodies, are usually extra susceptible to reinfection of SARS-CoV-2. Even if it's just a few weeks after the first infection. That's insane. Yeah, that's almost unprecedented, really. And so knowing this kind of stuff is really important, because if this finding ends up being confirmed, then we will have a tangible molecular explanation for why it's so important to quarantine elderly and immunocompromised individuals during a pandemic. Moving on to the second article, it's titled MERS, the New Coronavirus Explained. MERS stands for Middle Eastern Respiratory Syndrome. So it's very similar to the SARS virus that is responsible for COVID. In fact, as you can tell from the title, they're in the same family, the coronavirus family. Now, it's titled MERS, the New Coronavirus. So as you can tell, this article is already pretty dated. It's from 2015. Little do they know what would happen in a few years. So MERS disease is very similar to COVID-19 in a lot of ways. They both cause primarily respiratory distress, like coughing. They can cause pneumonia, fluid filling in your lungs. That's basically the mantra that goes with COVID-19 and MERS. But they can also cause a lot of other side effects, including kidney failure, liver failure in some cases. And both MERS and SARS-CoV-2 are transmitted from animal to human and human to human. And both of them seem to require close contact in order to spread. That's why you're hearing so much of this 6 feet of social distance rule. MERS, I decided to include this article not only because it's relevant to SARS, but MERS itself is a deadly virus. It's serious business, and even though the major outbreaks occurred several years ago, the virus hasn't gone away. Even I personally have seen several patients with MERS infections coming into the hospital while I've been working. But there is a gray lining. So the fact that so many viruses are related to each other, just like MERS and SARS are, the fact that they're related to each other gives us the opportunity to learn from past outbreaks and in order to deal with future ones. And so that's what a lot of researchers are doing. There are a lot of resources being currently dedicated toward finding more similarities between MERS and SARS and SARS-CoV-2 and SARS-CoV. There's a bunch of different types of coronaviruses. Because the more overlaps that we can find, hopefully, ideally, the more we'll understand how to treat both of these diseases. So viruses don't live in a vacuum. They're related to each other. This is going back to whether viruses are living or non-living. Viruses have evolved, so they've also evolved to be related to each other. And so we can exploit that by drawing connections between different families and different members of families. And finally, this third article is about a less heavy subject, the West Nile virus. So you've probably all heard of animal vectors before in biology, right? So that's basically what this is. A vector in terms of virology is like a middleman of infection. So West Nile virus, you can see the image includes a mosquito. West Nile virus ultimately comes from birds, but it spreads to humans through mosquitoes. So mosquitoes can get infected, can carry the virus when they make contact with the bird, and then they can infect us, the humans, once they bite us, once they suck our blood. And so here the mosquito is acting as a vector. And so recently, the West Nile virus was found in, I believe it's pronounced Galen, Ohio. It wasn't found in a human, it was found in a mosquito trap. Why these people were analyzing the genetic material of a mosquito trap, I'm not sure, I don't know. But in either case, they found West Nile virus, that's what's important, which could indicate a possible future quote-unquote outbreak. However, fortunately, unlike MERS and unlike SARS, West Nile virus isn't as pressing of a matter. I believe it's only like 1 in 5 infected people. That's people who are infected. Only 1 in 5 of them show major symptoms at all, and most people recover completely. So this is a good example of how not all viral diseases are necessarily world-shattering. For one, not all viruses are used for diseases. There are other uses of viruses that maybe we could save for another discussion. But even the viruses that do cause diseases, it's not like all of them are pandemic causing, you know. That being said, essentially all viruses are important, and more viruses are proving to have global pertinence, whether it be positive or negative. Viruses have the potential to kill humans and also save lives through vaccines, gene therapy, etc. So understanding how these nanomachines work and understanding how they can be manipulated is vital for improving the world that we live in. Viruses affect the whole world. They affect all of us. There are hundreds of millions of them. And that is the final note I want to end on. So thank you so much for listening, everyone.